The problem we are now facing is that we don't have enough cells to transplant into patients. And we don't have enough cells because we need several donors for each patient. So it is difficult to guarantee the viability and the quality of the material. So we've got to find alternatives to fetal cells. And to do so, we're focusing on the alternative of neural stem cells.
In this flask, I have human brain cells. These cells have been cultivated from cells taken from an aborted fetus that was 10 weeks old. They are cultivated as free-floating clusters and can proliferate for more than a year. In the beginning, there were only a couple of thousand cells in this flask, but they have since grown to several billion cells. Before I start manipulating the cells, I examine them under the microscope to check if they seem all right. If they have formed clusters and if they are not contaminated, these cells look fine. They have formed big clusters since their last passage 10 days ago.
This is how the neural stem cells grow in cell culture. They begin as little balls that grow bigger and bigger. This happens under the influence of growth factors that are added to the medium. And it's due to these growth factors that they grow continuously and divide week after week. The next step is to analyze what they can do after being transplanted into the brain. And we do that with rats. Here you can see a stem cell which has developed into a fully mature nerve cell with its characteristic cell body and processes. Here we see a cluster made of transplanted neural stem cells that have now formed nerve cells. They've migrated into the brain, and we can see the processes that have developed. These processes are essential for nerve cell function. We've not yet worked out how to make these cells produce the right kind of dopamine. Nor do we know how to get them to survive, grow, and function correctly in the same way as the normal dopamine cells. To solve these problems, we need extensive research, and it will take a long time, years, before we have the solutions.
Despite the immensity of the research work still to do, some scientists are already preparing to change gear. They are taking an even more radical and ambitious path by turning laboratories into small companies, which are preparing stem cells like pharmaceuticals, but for a hypothetical market.
The product is already down there, okay? It's down in the lab, but it will take us a little bit of extra time. The reason we think that this is important is we have to show the regulators that what we're putting in to the patient is the same cell as, is going into every other patient. And what I mean by the same cell, I mean it's a cell line derived from an original founder cell, multiplied zillions of times to make up a very large bank of cells, but genetically identical in every possible way, so entirely consistent. But the important thing is that the cells, it's a one-off treatment. It's not like you're taking a drug every week or every day or every 3 times a day. It's you have an injection of the cells, and that's it for the rest of your lifetime. There's a lot of work that's been done on using stem cells in animal studies, and so far there are very few reports showing that stem cells can be useful in humans. So the critical test is, companies like us. We're a small company. you know, we're taking risks working in a product like this because there's no guaranteed future for us in stem cells. We don't know whether they're going to work in patients, really.
Research with stem cells is far from finished. One major problem in therapy is the incompatibility between grafts and the patient, which leads to rejection. Some scientists hope to overcome this problem using a technique called therapeutic cloning. A technique which has the potential to produce tissues and organs that are 100% compatible with the patient.
Two things are needed for therapeutic cloning. First, a cell from the skin or muscle, or indeed any sort of cell from the future recipient patient. And second, an ovum from a consenting donor. The nucleus is taken out of the ovum and replaced with the nucleus from the recipient's cell. The fusion is completed by applying a weak electric field. The resulting cell will then start to develop. The development is stopped after 4 or 5 days and will never reach the stage of implantation into the womb. The embryonic stem cells are collected and grown in a test tube. They carry all the genetic information contained in the nucleus of the recipient and are therefore 100% compatible with all organs of his or her body. If we could learn to control the mechanisms by which stem cells differentiate into the various tissues, it would be possible to produce banks of muscle cells, bone cells, nerve cells, and indeed all cell types, made to measure. Cells ready to repair all the organs of every individual person with no risk of rejection.
The ability to repair man. An ancient dream come true? These techniques bring us hope, but some of these techniques raise ethical questions. To what extent do we have the right to use embryos, and even create embryos with the aim of healing the sick? Under which conditions can we use medicines that, like organ grafts, are composed of living human cells? Could the research on stem cells favor a slide from the cloning of tissues to the cloning of human beings? These questions are important to us all, because the answers will have a substantial impact on our future. We need to find a way to reconcile the freedom to do research and our desire to benefit from medical advances with respect for fundamental ethical principles. We must allow for a full and truly inclusive debate on how to handle such questions with dignity and a sense of responsibility.